Hello friends, today I am going to discuss about chemical foundations of physiology. In this particular topic, we talk about solutions, osmotic pressure and diffusion, the levels of organization of a language such as letters, words, sentences, paragraphs and so on can be compared to the levels of organization of the human body. Your exploration of the human body will extend from atoms and molecules to the whole person. From the smallest to the largest, there are 6 levels of organization. The chemical, cellular, tissue, organ, system and organismal level of organization. Chemical level is very basic level, can be compared to the letters of the alphabet and includes atoms, the smallest units of matter that participate in chemical reactions, and molecules, 2 or more atoms join together. Certain atoms such as carbon, hydrogen, oxygen, nitrogen, phosphorus, calcium and sulfur are essential for maintaining life. 2 familiar molecules found in the body are deoxyribonucleic acid, that is DNA, the genetic material passed from one generation to the next, and glucose, commonly known as blood sugar. Certain processes like metabolism, responsiveness, movement, growth, differentiation and reproduction distinguish organisms or living things. They are called life processes. When the life processes cease to occur properly, the result is death of cells and tissues, which may lead to the death of organism. Homeostasis is the condition of equilibrium in the body's internal environment due to the constant interactions of the body's many regulatory processes. In response to changing conditions, the body's equilibrium can shift among points in a narrow range that is compatible with maintaining life. For example, the level of glucose in blood normally stays between 70 and 110 milligrams of glucose per 100 milliliters of blood. Each structure from the cellular level to the systemic level contributes in some way to keeping the internal environment of the body within normal limits. An important aspect of homeostasis is maintaining the volume and composition of body fluids, dilute, watery solutions containing dissolved chemicals that are found inside cells as well as surrounding them. The fluid within cells is intracellular fluid. The fluid outside body cells is extracellular fluid. The extracellular fluid that fills the narrow spaces between cells of a tissue is known as interstitial fluid. The proper functioning of body cells depends on precise regulation of the composition of the interstitial fluid surrounding them. Let us see some of the important physiological aspects related to maintaining the homeostatic mechanism of the body. Solutions. A mixture is a combination of elements or compounds that are physically blended together, but not bound by chemical bonds. For example, the air you are breathing is a mixture of gases that includes nitrogen, oxygen, argon and carbon dioxide. A mixture composed of particles that are dispersed in another medium may be one of the 3 categories: solution, suspension or colloid. We emphasize on solution only. In a solution, a substance called the solvent dissolves another substance called the solute. Usually there is more solvent than solute in a solution. For example, your sweat is a dilute solution of water, that is called the solvent, plus a small amounts of salts, the solutes. Water is the most important and abundant inorganic compound in all living systems. Nearly all the body's chemical reactions occur in a watery medium. Once mixed together, solutes in a solution remain evenly dispersed among the solvent molecules. Because the solute particles in a solution are very small, a solution looks clear and transparent. In a solution, the individual molecules or ions of a substance, the solute, are small enough to dissolve in a liquid medium, the solvent. These molecules or ions are so small, that is less than 1 into 10⁻⁷, that they never settle out under the influence of gravity. The mixture is homogeneous, that is perfectly uniform in appearance. Molecules or ions of the solute in a solution cannot be filtered out. If a solution is heated vigorously, the solvent evaporates and the solute particles separate out as crystals. Therefore, solution is also sometimes referred to as a crystalloid. Sugar dissolved in water provides the best example of a true solution. When a solid is dissolved in a water up to the limiting concentration, it is called a saturated solution. There are 3 units of concentration that are particularly useful to chemists. These are molality, molarity and normality, and they are dependent upon mole unit. It is believed that there was a specific number that would represent the number of atoms or molecules in a mole of that atom or molecule. The weight of that unit, known as a mole, would be equivalent to the atomic or molecular weight of the atom or molecule in grams. Molality. A molality is the number of moles of solute dissolved in 1 kilogram of solvent. Be careful not to confuse molality and molarity. Molality is represented by a small m, whereas molarity is represented by an uppercase M. Note that the solvent must be weighed unless it is water. 1 liter of water has a specific gravity of 1 and weigh 1 kilogram. So, one can measure out 1 liter of water and add the solute to it. Most other solvents have a specific gravity greater or lesser than 1. Therefore, 1 liter of anything other than water is not likely to occupy a liter of space. To make a 1 molar aqueous solution of sodium chloride, measure out 1 kilogram of water and add 1 mole of the solute sodium chloride to it. The atomic weight of sodium is 23 and the atomic weight of chlorine is 35. Therefore, the formula weight for sodium chloride is 58 and 58 grams of sodium chloride dissolved in 1 kg water would result in a 1 molar solution of sodium chloride. Molarity. The molar unit is probably the most commonly used chemical unit of measurement. Molarity is the number of moles of a solute dissolved in a liter of solution. A molar solution of sodium chloride is made by placing 1 mole of solute into a 1 liter volumetric flask. Now, taking data from the example above, we will use 58 grams of sodium chloride. Water is then added to the volumetric flask up to the 1 liter line. The result is a 1 molar solution of sodium chloride. The equation goes like this: molarity is equal to 1 mole into 1 liter. Normality. There is a relationship between normality and molarity. Normality can only be calculated when we deal with reactions, because normality is a function of equivalence. The example follows uses potassium hydroxide to neutralize arsenic acid. By studying the reaction, it is possible to determine the proton exchange number to determine the normality of the arsenic acid. Look at the equation. Arsenic acid plus 2 KOH, that is potassium hydroxide, give rise to potassium arsenate K2HaSO4 plus 2 molecules of water. Equivalent weight is equal to molar mass per hydrogen ion per mole. Equivalent is equal to mass of compounds per equivalent weight, and normality is equal to equivalence of X per liter. And the part that is of interest to you is that normality is equal to molarity into N, where N is equal to the number of protons exchanged in a reaction. You probably remember that when a hydrogen atom is ionized and loses its electron, you are left with only a proton. So, a hydrogen ion is basically a proton. Let us assume that we have 0.25 molar solution of arsenic acid and want to determine the normality of it. If it participates in a reaction, it goes like this: arsenic acid plus 2 KOH gives rise to potassium arsenate plus 2 molecules of water. When arsenic acid is neutralized by potassium hydroxide, arsenic acid provides 2 protons to form 2 molecules of water. Note that arsenic acid has 3 hydrogens, but potassium arsenate only has 1 hydrogen. That means that 2 protons were exchanged. Again, a normality is equal to molarity into N. Remember that normality of the solution is 0.25 mole arsenic acid and there were 2 protons exchanged, 2 equivalents per mole. That is why 0.25 moles arsenic acid into 2 equivalents of arsenic acid into 1 liter of arsenic acid is equal to 0.50 equivalents of arsenic acid into 1 liter of arsenic acid becomes 0.5 normal arsenic acid solution. So, in short, while there is a relationship between the normality of a solution and the molarity of a solution, the normality can only be determined by examining reaction, determining the proton exchange and multiplying molarity by that number. Normality is particularly useful in titrations calculations, where N is equal to normality, V is equal to volume, A, that is the substance on the left of the equation involved in proton exchange, and B is equal to substance on the right of the equation involved in proton exchange. That is, N A V A is equal to N B V B. Percentages are easy to calculate because they do not require information about the chemical nature of the substance. Percentages can be determined as percent by weight or percent by volume. Let us say percent by weight. To make up a solution based on percentage by weight, one would simply determine what percentage was desired. For example, a 20% by weight aqueous solution of sodium chloride and the total quantity to be prepared. If the total quantity needed is 1 kg, then it would simply be a matter of calculating 20% of 1 kg, which of course is 0.20 sodium chloride into 1000 gram per kg is equal to 200 gram sodium chloride per kg. In order to bring the total quantity to 1 kg, it would be necessary to add 800 gram water. Percentage by volume. Solutions based on percentage by volume are calculated the same as for percentage by weight, except that calculations are based on volume. Thus, one would simply determine what percentages was desired. For example, a 20% by volume aqueous solution of sodium chloride and the total quantity to be prepared. If the total quantity needed is 1 liter, then it would simply be a matter of calculating 20% of 1 liter, which of course is 0.20 sodium chloride into 1000 milliliter per liter is equal to 200 ml sodium chloride per liter. Percentages are used more in the technological fields of chemistry, such as environmental technologies, than they are in pure chemistry. Using the information above, it is possible to calculate concentrations of solutions and make up solutions of desired concentrations. Osmotic pressure. It is necessary to understand the process of osmosis before we talk about osmotic pressure. Osmosis. If two solutions of a different concentrations are separated by a semi-permeable membrane, the solvent molecules move across the membrane. In living system, the solvent water, which moves by osmosis across plasma membrane from an area of higher water concentration to an area of lower water concentration. Another way to understand this idea is to consider the solute concentrations, in which water moves through a selectively permeable membrane from an area of lower solute concentration to an area of higher solute concentration. Osmosis occurs only when a membrane is permeable to water, but it is not permeable to certain solutes. A simple experiment can demonstrate osmosis. Consider a U-shaped tube in which a selectively permeable membrane separates the left and right arms of the tube. A volume of a pure water is poured into the left arm and the same volume of a solution containing a solute that cannot pass through the membrane is poured into the right arm. Because the water concentration is higher on the left and lower on the right, net movement of water molecules, that is called osmosis, occurs from left to right, so that the water is moving down its concentration gradient. At the same time, the membrane prevents diffusion of the solute from the right arm into the left arm. As a result, the volume of water in the left arm decreases and the volume of solution in the right arm increases. You might think that osmosis would continue until no water remained on the left side, but this is not what happens. In this experiment, the higher the column of solution in the right arm becomes, the more pressure it exerts on its side of the membrane. Pressure exerted in this way by a liquid known as hydrostatic pressure forces water molecules to move back into the left arm. Equilibrium is reached when just as many water molecules move from right to left due to the hydrostatic pressure as move from left to right due to osmosis. The pressure exerted by the osmotic process is called osmotic pressure. It can be regarded as a form of suction drawing water from a weak to strong solution. The osmotic pressure of a solution is proportional to the concentration of the solute particles that cannot cross the membrane. The higher the solute concentration, the higher the solution's osmotic pressure. If the pressure is applied to the fluid in the right arm of the tube with the help of piston, consider what would happen. With certain pressure, the volume of fluid in two arms could be restored to the equal and the concentration of solute in the right arm would be the same as it was at the beginning. The amount of pressure required to restore the starting condition equal the osmotic pressure. So, in our experiment, osmotic pressure is the pressure required to prevent the movement of water from the left arm into the right arm. Notice that the osmotic pressure of a solution does not produce the movement of water during osmosis. Rather, it is the pressure that would prevent such water movement. Normally, the osmotic pressure of the cytosol is the same as the osmotic pressure of the interstitial fluid outside cells. Cell volume remains relatively constant because of osmotic pressure on both sides of the plasma membrane is same. When body cells are placed in a solution having a different osmotic pressure than cytosol, However, the shape and volume of the cell change. As water moves by osmosis into or out of the cells, their volume increases or decreases. A solution's tonicity, what we call a tension, is a measure of the solution's ability to change the volume of cells by altering their water content. When a cell is placed in a solution, water will pass in or out of it according to the osmotic pressure of cell contents and the surrounding medium. From the biological viewpoint, solutions can be grouped into three classes: isotonic, hypotonic and hypertonic solution. Isotonic solutions which have the same osmotic pressure as that of the cells. The concentrations of solutes that cannot cross the plasma membrane are the same on both sides of the membrane in this solution. For instance, 0.9% sodium chloride solution called a normal saline solution is isotonic for red blood cells. The plasma membrane of red blood cell allows the water to move past in and out, but it is impermeable to sodium and chloride ion, the solutes. Water molecules enter and exit the red blood cell at the same rate. So, RBC maintain their normal shape and volume. When RBCs are placed in a hypotonic solution, a solution that has lower concentration of solutes than the cytosol inside the RBCs, in this case, water molecules enter the cell faster than they leave. As a result, red blood cells swell and finally burst. The rupture red blood cells in this manner is called hemolysis. A hypertonic solution has a higher concentration of solutes than does the cytosol inside red blood cells. One example of hypertonic solution is 2% sodium chloride solution. In hypertonic solution, water molecules move out of the cells faster than they enter, causing the cells to shrink. Such shrinkage of cells is called crenation. Osmosis has biological significance. Like in living cells, water is distributed and redistributed across membranes by osmosis. Continuous osmotic exchange goes on between blood, tissue fluids, tissue cells and lymph, and osmosis plays a significant role in the regulation of urine formation as well. Diffusion. Diffusion is a passive process in which the random mixing of particles in a solution occurs because of the particles' kinetic energy. Both the solutes, the dissolved substances, and the solvents, the liquid that does not dissolving, undergo diffusion. If a particular solute is present in high concentration in one area of solution and low concentration in another area, solute molecules will diffuse towards the area of lower concentration and they move down their concentration gradient. After some time, the particles become evenly dispersed throughout the solution and the solution is said to be at equilibrium. The particles continue to move about randomly due to their kinetic energy, but their concentrations do not change. For example, when a piece of dye is put into a container filled with water, the color is more intense in the area nearest to the dye because its concentration is higher there. At increasing distance, the color is lighter and lighter because the dye concentration is lower. After some time, the solution of water and dye will have a uniform color, because the dye molecules and water molecules have diffused down their concentration gradients until they attain a state of equilibrium. Several factors influence the rate of diffusion of substances across plasma membrane. First is the steepness of concentration gradient. The greater the difference in concentration between two sides of the membrane, the higher the rate of diffusion. Second is the temperature. Heat increases the intensity of movement of particles. Third is the mass of the diffusing substance. The larger the mass of the diffusing particle, the slower its diffusion rate. Smaller molecules diffuse more rapidly than larger ones. Surface area. The rate of diffusion is fast when surface area available is more. Diffusion distance. Diffusion occurs very fast when distance through which molecules has to travel is less.
Now, we have come to know that all bodily chemical reactions occur in some type of solution and have learnt about the nature of solution and how it reacts with various compounds. When two solutions with different concentrations are put together and separated by semi-permeable membrane, the molecules start to move from the area of higher concentration to lower concentration in order to achieve equilibrium by osmosis, and finally it is influenced by osmotic pressure. Diffusion is a process by which molecules move down their concentration gradient without change in concentration. So, in this section we have learnt about solutions, osmotic pressure and diffusion. We will discuss some other very important physiological processes like pK, pH and buffers in next lecture, that is chemical foundations of physiology.